So I sort of began to see that actually what note-taking should be, and I think I felt this for quite a long time, is literally that. You take notes, right? These are reminders to yourself of what the information was that you came. You have chapters in a book that you read. You have the internet, which you read. You have every resource I never had available to me. When I want to know something, I look it up. Here, the purpose should be you take notes, literally notes. You make yourself reminders. Oh yeah, he talked about this, I should look this up. He talked about this. You should try to listen. And you know, it's funny is that I've had students who go, no, it won't work. And then eventually they crack under the pressure and they just sit there. And I had one of my students who said, you know, I'm not smart as you, blah, blah, blah. I said, just sit in the classroom and listen. And he finally broke and he just sat in the classroom and listened. And he did very, very well in the class. And I was really surprised. I finally got him cracked. He went to medical school and, you know, he realized what I was trying to get to him because you think I'm teaching you a lot of information now. This is, you know, one course here at Bio 303, you would learn the entirety of Bio 303 in 4 weeks while learning pharmacology, anatomy, physiology, 3 or 4 other courses with tremendous amounts of information. And, you know, I had a lot of friends, I assume a lot of you are pre-med, I have a lot of friends who would be crying to me because they're on the brink of failing courses. And this person also, he wasn't crying, but he says, you know, I'm on the brink of failing, except for molecular biology that you taught me, I'm getting an A in that. Right? Because, you know, I got him to internalize the information, so all he had to do was bring the new information on top of the old information. He didn't have to learn from scratch. And that's really what you're trying to do. So I'm gonna try very hard to get you to sort of grasp what I'm talking about. I tend to refer to this lecture or this sort of intro as the central dogma revisited. We'll see where I go with this and see what you guys take away. Yes, you had a question.
Do you think it's beneficial for the way that you teach in the class to read the textbook beforehand?
So I'll say yes and no. I mean, and the reason I say that is I hate textbooks. I find most textbooks inaccurate, flawed. When I taught Bio 302, I was given the textbook because it was a split class. So I was forced to teach from the textbooks. I was not the lead in the lecture, and so I was forced to use the textbook that the other professor used. And I would go through it and go, oh no, this is wrong. Oh, I really don't want to teach it like this. Oh, I have to take the PowerPoint slides and rechange them because they're not right. And I just got very, very frustrated. You know, one of my favorite examples, you're taking cell biology now. I don't know if you've heard of what's called a 7' membrane receptor. One, two, three, 4, 5, 6, 7. So 7 transmembrane receptor. Here's a lipid bilayer. Right, so this is the way almost every book represents it. Lipid bilayer, extracellular, intracellular. Right, cell biology class. I don't know if you learned about GPCRs yet. 7-PM protein, 7 transmembrane, also referred to as G protein coupled receptors. Something I teach in grad cell biology, so important, but just sort of give you an ideology of why I hate textbooks. So again, 1, 2, 3, 4, 5, 6, 7. This is the C terminus, the N terminus. Right, this is sort of a classic descriptor of this type of protein. It's also called serpentine because they're like snakes. And if you may not know, there's a favorite receptor, beta-2-adrenergic receptor, which binds ligands like isoprenolone or epinephrine, and they do things and they change the physiology of your cells. And so, so it's one of my favorite examples, and you might see a ligand like this, going like this. ligand that will bind to the receptor. All right, so now let's take a look at what, again, this is just your listening. This is perspective. You're just listening. You're not taking notes. You're just listening and saying, what am I trying to tell you? So it turns out, if you look at a 17 receptor, it's usually about 300 amino acids. But if you actually look top down on a receptor in a three-dimensional structure, it looks like this. 1, 2, 3, 4, 5, 6, 7. Because these are alpha helices that compact down into each other. So this what's called the schematic representation, which is all over your textbook, is just not only incorrect, but gives you the wrong sense of the structure of the molecule. And then it's worse. Remember I showed you, there's some ligand here that binds it. Again, I told you 300 amino acids. Somebody tell me how big epinephrine might be relative to the size of an amino acid. Somebody must have want to say something. I do ask questions of the class, and I wait for answers. So somebody might want to, yes?
Probably a lot larger.
Your one says a lot larger. So it turns out, anyone know how many, what's the size of a typical amino acid? Right, so let me just say, you're taking cell biology, you should be way beyond molecular genetics where you learned all this. You'll see this is why I revisit some of these things, because people already have the wrong groundwork for incorporating cell biology. So how big is an amino acid in Dalton? Without looking it up on your phone.
Mm-hmm.
What's the average weight of an amino acid? No one knows? Come on. I'm going to go tell Dr. Eckhart and Dr. Eliza. No one in the class. And I'm going to tell Adrian that no one in the class can tell me without looking it up on their phone, what the average size in Daltons of an amino acid is. It's an important number. I mean, this is really serious. No one knows. Tatiana, you don't know. This is in my lab. No one really knows. No. How about a guess? How many Daltons? 100. 110 Daltons is the average. And that's 0.1 kilodaltons. So it turns out a ligand for a receptor like this, which binds somewhere around here, I'll draw a different color, actually is the size of about 1 amino acid. And as I told you, so 1 amino acid, this thing is actually 1 amino acid in size. But you see how it's drawn. It's drawn as this hulking thing of like representation of hundreds of amino acids in size. You can't actually draw it in a textbook because it'd be, so remember, this is a lipid bilayer. How many amino acids needed to pass the lipid bilayer? 20, roughly 20, 19, 20. So that means that if you carve this up into 20 equal parts, I can't even do it, it's probably the size of a pinprick, that amino acid in this representation. You will be hard pressed to find a textbook draw it as a pinprick, which is the true representation. Again, you're not here to, it's more than just you're learning it's a ligand receptor, you're learning perspective. This is, I think, the ultimate goal of learning biology. I learned biology the way I'm sure all of you guys learned it before, which is go memorize this, go learn this. And the day I walked into the lab, I was like, oh wow, I know nothing. I know absolutely nothing. I can't do anything. I have to reread everything. I mean, I was so embarrassed by that. Although I was a chemistry major, biochemistry major in college, when I graduated college, I started taking biology courses again. Didn't know anything. I started from scratch, which is really embarrassing. Absolutely from scratch. Even though I had learned stuff, I knew I learned none of it in perspective. Start over. Since, not everyone will do something like that. I'm not glutton for punishment like that, but, I realized, if I ever wanted to learn biology, I had to start somewhere. So this is what I mean by your understanding or your sort of internal representation of knowledge may not be exactly the way I would like it. So that's why I call this lecture Central Dogma Revisited. Because you've probably all heard about the central dogma, you all know the components, relatively speaking, of the central dogma. But, you know, sometimes I think that those memory engrams are a little bit flawed, and so I try to fix those along the way. I used to give a test when I get a few quiz questions along the way. I probably won't do that to you guys, because I used to give quiz questions just for fun, just to see the depths of foreknowledge of students. And, you know, it was so painful to see that I stopped doing that, because I managed to figure out, okay, we're not in a level playing field. There are people who really don't understand stuff, and so instead of just enjoying to see the crazy answers I would get, I decided, okay, let's just clean the slate and start over. And so that's why. Okay, so you guys all took Bio 2 and 3, right? And, right, and so I think you guys all know that the central dogma is essentially a coding dogma, right? It's how you code information. Right? That's what it is. And so the central dogma says you have DNA, which is double-stranded. That DNA provides a template for making RNA. And that RNA provides a template for making protein. That's the basic one. Protein, and RNA. You know, I'm not looking behind me. So that's the basic thing, and you probably have learned, and tell me if you haven't, that DNA can lead to RNA. RNA can go back into DNA. RNA can go into protein. DNA can go back into DNA. Right? So you can use DNA to code DNA, you can use RNA to code DNA, RNA to code RNA, RNA to code RNA. And everyone talks about, like, this is not possible. We'll revisit that a little bit later, right? That's what you've all been taught, right? One-way information, once you get to protein, that's it, it's terminal. It's interesting, however, right, that every other aspect can do the coding, but not the protein. And I have to say, the reason why I say I sort of half jazz is interesting, because I always think when I have to teach people something, I actually have to relearn it myself and think, well, why is it that protein doesn't go backwards? And I think through, right, I'm forced to teach you guys, so I'm forced to learn it myself. And then I ask the hard questions. And actually, it's kind of funny is that, you know, I'm 54 years old. And, you know, sometimes I think, oh, well, I really know it. And then I start reading something and I go, really? That's how it's being taught? Really? And so then I start actually looking up the literature on why it's being taught this way. And I really think through, well, really, am I going to really teach it the way the book is teaching it? And is this really the right way to do it? Remember, this is, I think, the most important thing you should remember for you guys in this generation. You guys have all the information at your access. I had nothing. I had absolutely nothing. I had old textbooks. You know, it used to take 5 years or 10 years to get rid of a textbook. I mean, the information was completely outdated when I was learning. It was awful. Awful. So, you guys have it made. You have it on your phone. Right? This is how great it is. Okay. So this is sort of the central dogma of biology, and we're going to dig a little deeper. So, again, this is not patronizing, but I like to start everyone from scratch. So we're going to start from scratch. And by the way, I study the sense of smell, olfaction, which is a neuron. But actually, I think all cells are cells. I don't think of myself as a neurobiologist, I think of myself as a cell biologist. I study olfactory neurons. But I'm not a neuroscientist, I'm not a smell biologist, I'm studying the property of this particular type of cell. And that's sort of how I also want you guys to understand it. So, you know, if you have a cell, most cells, except for mature or red blood cells, have a lipid bilayer. Again, I'm sure all you guys know this. And if you look inside the cell, of course, there's a nucleus. And of course, there are lots of other pieces in the cell. There's the ER, there's Golgi, right? All this, I'm sure you guys learned this. And there are lots of little vesicles inside lysosomes. There's actually this peculiar little structure, which I'm ashamed to say I think I didn't learn until very, very late in life, after college. This thing called mitochondria. I was actually really surprised that this had its own nucleus, had its own DNA. That DNA is a 16 kilobase circle. Actually, there are a lot of mutations in that plasmid DNA or that circular DNA that can get you quite sick. And so this little mitochondria here differs from cell to cell, meaning that the exact number of mitochondria is not the same in every cell. And depending on the mutation of that circular piece of DNA inside, some cells are hit harder than others. So there's some eye diseases, there are some kidney diseases, whole host of different diseases depending on the luck of the draw of how many mitochondria you inherited in that tissue. Because when cells divide, mitochondria go with it, and they also have a certain division rate. These things divide, have their own program to divide. But one thing that's interesting about mitochondria and mitochondrial DNA is that none of this functions without host proteins going into the mitochondria. Okay? Even though it has its DNA, it's lost the ability to be self-autonomous, not self-autonomous. Curious little structure. I was really, really surprised by that as I was starting to learn deeper about genetics and biology. So, and then of course the nucleus. So we're going to start today with the nucleus, and we're going to work inside out. Actually, I think most of my lectures are around the nucleus, but we'll see. We'll move in and out of the nucleus. All right. So let's look inside the nucleus, and again, all of you guys have probably learned this. Humans have 23 pairs of chromosomes. You know, I write this just to see if you'll write it. Just sort of a little game I play to see how much can I expect you to believe that. Of course, this is only true for women because we have 22 autosomes that are paired, and women have XX. Of course, men are 22 autosomes and that's why. So men are not 23 pairs of chromosomes, but you've all been taught that, so you all regurgitate it. It's not true. Actually, 24 kinds of chromosomes. It's just one of those regurgitation facts. Mice, which you probably learned a lot about, have 19 chromosomes plus X and Y. You'll see why this is relevant in a little bit. But this, for example, is roughly about 3.9 times 10 to the 9th base pair. And actually, mice also have about 3.9 times 10 to the 9th base pair. But somehow those chromosomes differ, right? We have essentially 24 kinds of chromosomes. We have 21 kinds of chromosomes here. Information must be compacted differentially. And this is you'll sort of get to see how I like to think about biology, is that you should be able to figure that out on your own. You should not need me to tell you this. You should be able to instantly see, okay, the way DNA exists in humans and mice is fundamentally different because it's compacted differently. you shouldn't have to be told that. You should be able to go, there's a disconnect here. That's how I read a lot of biology. I see two facts and I go, no, this doesn't quite make sense. Okay, so we'll get to that, the importance of that in a second, because again, I'm sure you guys have all learned. Please tell me if you've learned this. There's a P and a Q arm to a chromosome. Everyone agree? And so there's a centromere here, and a telomere here. Right, everyone's on the same page, right? Unfortunately, it's only true for human chromosomes, it's not true for mice. So it turns out that mice have telocentric telomeres. So this is mice, they have no P arms. They only have Q arms. And this is centromere and telomere. So they're really different than human DNA, right? This is the centromere, I'll just highlight it, centromere, and then there's a little telomere. So now you know the DNA is really distributed very differently. Okay? And we'll get back to that in a moment. But you see, one begets the other, right? Once you know this, you can sort of begin to say, well, maybe the P and Q thing is not really identical, right? Because we know there has to be a different distribution of DNA. Those are the kind of steps you're going to make. Okay, let's continue. So, and then this actually I also learned quite late. I'm a little embarrassed to say that in humans, right, we have chromosomes 1 through 22. How are they numbered, the chromosomes? Yeah. Size. So chromosome 1 is bigger than chromosome 22. And if you know, for example, one of the most hereditary genomic anomalies is trisomy 21. And again, once you understand that 21 is a small chromosome, not as small as 22, you can understand that you will never see a trisomy 1. That can't exist. The only reason trisomy 21 exists is because there's less genomic information that's being triplicated in a cell. Right? Just logic. So you'll not see a trisomy 1, not see a trisomy 2, 3, 4, 5, all the way down. Does that make sense to everyone? So in fact, the common ones are trisomy 19, 21, forget which one, maybe I think you get some partial 8 trisomies. You don't have to always make the entire chromosome trisomatic in order to get inherited disease. And again, so we'll go back here again. 24 versus 21. Now that's the same question. I always think this is fun. So in mice, 1 to 19, how are the chromosomes numbered? Side, still side. So now you see the really big disconnect coming our way. Okay, we said there are 3 less chromosomes roughly. They're numbered by size. So does human 1, is human 1 the same as mouse 1? Does it contain the same information? No. In fact, the information's completely jumbled up. No chromosome except for the Y. Maybe a little bit the X. But the Y carries, Y is fairly stable. It can't recombine so easily. So Y's between mouse, rat, human, whatever, pretty similar. They carry one important gene, the Y, does everyone know? SRY, the sex determinant gene. It's the main gene. In humans, there are about 6 genes. But I think none of them are lethal except for SRY. Well, SRY is not technically lethal, you just become female. So it's not exactly lethal. That was not a joke, by the way. So DNA is distributed somehow differentially from human to mouse. And so we refer, so for example, if you look at human chromosome 1, you would say, okay, and we're comparing it to mice now. You might say, well, this portion here, I found on mouse chromosome 3, and this portion here, I found on mouse chromosome 2, and this portion here, not good. I'm using that black. Goes to chromosome 5, and then you can get really weird. And okay, we'll just continue on our way. This part is on chromosome 8, et cetera, et cetera. And what's more interesting is, so now let's draw out mouse chromosome 3. So let's say this is mouse chromosome 3. Right, and this is, let's say this is the edge of the T arm. I make up stuff like this and people get frustrated. That's why I said, listen, stop copying, listen, right? Because I'm just drawing things off the top of my head just to make a point. I'm not drawing this to make you memorize it. So let's say this is the PN. There's no PN in chromosome 3 of mouse. So let's just arbitrarily call this 1 in 10. Just one position to 10. That doesn't mean 1 in 10 is exactly 1 to 10. It could be 10 to 1. It could be the whole chromosome's inverted on 3. Or that whole portion is inverted. And here, let's call this, again, 1 to 10, just for argument's sake. And on chromosome 2, that portion is also at the beginning of the chromosome, but it's also 10 to 1. Right? It's completely jumbled up. And what we mean by this portion is this, we call these pieces, and this is one of the first words I want you to remember, syntenic, or synteny. You find synteny between parts of the chromosome, meaning they're not identical. As I told you, we're both have roughly the same amount of DNA. I think there are a total of 50 genes that go either direction. You find them in mice and not humans, and 15 humans, not mice. Of the 20,000. So most of the genes are identical, at least in roughly function. You can find a correlate from one to the other for roughly almost all the genes except for a handful. And we call these pieces of homology syntenic, or shown synteny. So let's continue. So synteny implies a very important word called orthology. They're orthologs. So for example, if there's beta-2-adrenergic receptor here, there's certainly a mouse ortholog that has beta-2-adrenergic receptor. They are orthologous to each other because, in part, they are syntenic. Right? They are collinear in the chromosome. That means there's a gene. So if you look on human, for example, and you see beta-2 here, there might be a gene X and a gene Y here next to it. And now you go to mouse, and you find beta-2, and you also find an X and a Y homolog, or an ortholog. These are syntenic. These all show synteny. and we refer to the syntenic gene on one chromosome to the other ortholog, meaning that they are historically what we imagine to be the same functioning component of that DNA. There are two other words that are related to orthologs that I think are important for you to know, that are thrown out a lot, but I want to, again, like I said, sometimes I tell you to write down a word, remember, it's not because it's gonna be on the test, remember, I'm testing you on logic, I'm not going to test you on vocabulary. Vocabulary is for someone else to test you on. I'm not gonna test you on vocabulary. So, there are two other words that you should know, it's called homolog and parallel. So, there are actually, there's beta-2 adrenergic receptor, and actually, there are two homologous genes, beta-1 adrenergic receptor and beta-3 adrenergic receptor. They're similar functions, they bind up in similar structure. They actually have very, very similar amino acids, very, very similar nucleotide sequences. We refer to those as homologs. Some would be, if it's very poor homology, we say they're distant homologs, right? So you could also actually argue, and some people use this sort of terminology, some people say, well, they're human and mouse homologs, yeah.
For the beta-2 genes that you talked about in human and mice, why is it important to have them in between the same genes for them to be considered syntenic?
Because synteny implies parallel distribution on a chromosome. So, for example, if there was another, if for example, beta-2 was not here, but X and Y were, beta-2 was just alone somewhere else, you would not refer to this beta-2 piece as being syntenic to this.
So they might be in different chromosomes, but they still are in the same.
They are orthologous, but they're not syntenic. Because it turns out genes can move all over the place. And sometimes actually beta-2 is one of those examples. Remember we talked about way back here, everything comes together. So it turns out beta-2 adrenergic receptor is a splice gene that went back into the DNA as a splice gene, and then acquired a promoter and acquired novel function. And so these RNAs that get back translated into DNA that usually are brought back into the genome as viruses become a new gene. And that happens all the time. It happens in viruses all the time. You have CSARC, which is a gene that's in your body, CSARC, and BSARC, which is the spliced version in viruses that re-enters your body, but now re-enters in a place somewhere else. There's a fully spliced gene with a promoter, and it goes and it does some nasty stuff to you. So you know, as I said, it's sort of this is information about how to think about these. And then there's this unique word here that you guys really don't have to worry about too much. But since I'm teaching how to use words, I'm going to teach you what a paralog is. Does anyone actually know what a paralog is? No one learned this in genetics. Yes?
Even in the evolutionary class, a parallel is when one gene gets copied and it starts acquiring different mutations.
So then, are beta-1, beta-2, and beta-3 paralogs?
No, that's what I missed.
Right, because these are all similar to each other. They've certainly developed by duplication. Yeah?
I think when the gene is duplicated, but then it's separated by speciation. Like hemoglobin is an example, I think. Like alpha and beta chain.
those are actually gene duplications, those are not paralogs. But in fact, beta globin has, I don't remember all the forms, there's gamma 1, beta gamma 2, beta epsilon, beta fetal, and I think there's a delta 2. They're all sitting right next to each other. These are paralogs. These are nearly identical. We refer to those as paralogs. Because in fact, in some way, a paralog can't actually tell which one is doing the work. There are two of them, right next to each other. In fact, this is one of the most common ways that we probably have undiagnosed disease in our body, is that we have a gene duplication, one extra copy, and one extra copy is doing nasty stuff to us, just like trisomy. We don't have a mutation, just an extra copy, and that extra copy changes the balance. I always like to think of a cell as like an orchestra playing. You add an extra piece to that orchestra, it's not so good for the orchestra. So these are paralogs. These are very, very recent gene duplications, where almost you can't even ascribe a difference to them. Sometimes you can, but even if you could, we refer to those as paralogs, we refer to these as homologs, and if you were to look synthetically across, the human beta gamma 1, beta gamma 2, beta epsilon, beta fetal, beta delta, I think that's 3 of them. These are orthologs. And these are orthologs, and very often they are synthetic. They almost always are synthetic. I think these are useful to understand some of the core biology. So we're going to spend the next, we have what, 6:50, right? 25 minutes learning a little bit more about genes. So as I said, we're going to go all the way in, and then we're going to come all the way out. So again, most of you have learned this. I think it's extremely important to understand what the definition of a gene is. So, who wants to tell me the definition of a gene? You all took 203, right? Yep. A unit of genetic information. So by that definition, is a promoter a gene? That's a unit of genetic information. Yep.
I'm guessing. Unit of genetic information that codes for a protein.
Okay, so she wants it to make a protein. This goes back to the old again, I'm not criticizing, this is what I learned too. One gene, one protein. So there are lots of RNAs that function as RNAs.